Okay, our next speaker is Luke Kosinski, and he's going to be speaking about translational errors per structural order from potential polypeptides, facilitating future innovation.
Thank you all for coming. Can you all hear me?
No.
No. Did you switch the mic on? Is that better? Oh, Jesus. Thank you all for coming. I'd like to start by framing this question I have on the board, what are the consequences of translational error, in terms of a specific type of translational error that I'll be focusing on for this talk. So, typically, what I have here is an mRNA, and got a 5' 3' end. The blue region is meant to represent the reading frame, which ends with a stop codon. So typically what happens is a ribosome will bind, translate that open reading frame, and dissociate, creating a particular type of protein product. However, there's an error that can occur where the stop codon gets read through into that 3' untranslated region, which adds on a C-terminal extension to the existing polypeptide, creating a different read-through product, like so. And I purposely chose two different looking proteins here because that read-through product can change what the polypeptide result is quite a bit. So the question here that I'm looking at is, what are the consequences of making that read-through product? So the consequences can change depending on how frequently the errors occur. So if you have errors that are very rare, they tend not to have very much effect. You might have some errors which never have any effect. That's the really small bar on the right. Then you might have a bunch of errors that might have a very small effect, but they almost never happen, so they don't have much of a chance to really impact the cell. However, if you increase the rate, then you expect the magnitude of effect of the ones which can have an effect in the cell to grow. And this is what I'm looking at here, in theory, this should also expose these sequences to selection, allowing selection to purge some of them, and since these are relative frequencies, increasing to 9. And when you have even higher read-through rates, again, you expect the magnitude to increase, but it should also expose those sequences to selection, allowing for the deleterious sequences to be selected against and the benign sequences to remain. So what makes a non-coding sequence benign? We might think about what makes a non-coding sequence harmful in terms of if it's interacting with existing protein domains, messing up their function, or if it's forming dangerous aggregates like amyloids, which can be toxic to a cell. So what would be benign should be something which doesn't interfere with existing protein domains and doesn't form these dangerous aggregates. So there might be a lot of reasons why that could happen, but one possible reason is intrinsic structural disorder. So what I have here is a protein where the middle of the protein has some ordered sections that you expect to see in biochemistry, like the alpha helix and the beta sheet. But the tails on each side are disordered. They don't have a set structure. They tend to associate with the cytosol because they're hydrophilic, which also means they tend not to get into the buried hydrophobic residues that allow the protein to fold properly, so it doesn't mess up the existing folding, or at least in theory, and the existing protein domains. And it also is hydrophilic, so it tends not to form dangerous aggregates. And the nice thing about intrinsic disorder is that I can measure it using a program called IUPRED from just the amino acid sequence alone. So I don't need anything else. I can just use the amino acid sequence and get the amount. I get an estimate for the amount of inherent disorder for an amino acid sequence. So in order to test this idea that that ISD would make a non-coding sequence benign, what I have here is a plot of all the different genes from Saccharomyces cerevisiae compared for their ISD and split up by different gene regions. So on the x-axis, I have different coding regions for the C-terminal, the middle, and the N-terminal region. And then I also have the non-coding 3' untranslated region. And on the y-axis, I have the intrinsic structural disorder as measured by IUPRED. And as you can see, the open reading frame regions, the coding regions, have relatively high intrinsic structural disorder compared to the 3' untranslated regions, which, as long as they're not translated, can have they don't have to be selection acting on them. So they could potentially be deleterious if they were translated. So the idea here is that when you have something more gene-like, that can't be particularly harmful to a cell, whereas the free common translated region, as long as the error rates are very, very low, they don't have to follow any of the things we'd expect from a polypeptide that is normally expressed inside a cell environment. So my hypothesis then, for what I'm looking at, is when we have, that's just the average over the whole genome, but if we have some examples where there's higher intrinsic structural disorder, that should also correlate with more read-through, so more errors would expose those sequences to selection and allow them to purge the really the lower ISD. Or if you have less intrinsic structural disorder, that can also correlate with lower read-through error rates. So this is just words, and I should state that the causation here could go either way. On the one hand, if you have more errors, that could expose sequences to selection more frequently and reduce the intrinsic structural, or increase the intrinsic structural disorder, sorry. Or if you have intrinsic disorder already present in that untranslated region, it could be that you don't need to evolve really robust translational proofreading machinery that would prevent high error rates. So this could be measured using, in order to measure read-through, I've got a measure for ISD, but in order to measure read-through, I'm using a technique called ribosome profiling, which gives a snapshot of translation. So if these are ribosomes on transcripts, what the ribosome profiling method does is it uses the nuclease to chew up the free nucleotides. You can use centrifugation to get the ribosomes and get the ribosome protected fragments. So the nucleases won't be able to chew up the fragments that are protected by the ribosomes. And then you can go back to the annotated genome and map these back to the genome and see where they align, and see where exactly the ribosomes are. So the more ribosomes you have, the more relevant translation you should also have. So many of these reads will map to the start codon, near the start codon, others will map to other parts of the open reading frame. And sometimes you may have some, and these are the ones I'll be looking for, that map to that 3' untranslated region. So the more reads in that 3' translated region, the more read-through is occurring. And so what I'm looking for is a correlation between the number of reads there and the amount of intrinsic structural disorder for those untranslated, for that 3' untranslated region. Okay, so I did find a correlation between those, and I think, I guess I should say that I realize this looks kind of like a cloud of points, but the important thing to note here is the biological significance of it. So part of what's difficult to see is that there's so many points, there's kind of points on top of points. But if you look on the y-axis has got the 3' untranslated ribosome profiling reads, and the x-axis has the intrinsic structural disorder. And if you look at the going from the low intrinsic structural disorder to the high disorder, that goes from about 2 or so ribosome profiling reads on average and moves up to about 6 or 7 ribosome profiling reads on average. So that's a 3 to 3.5-fold increase in read-through. Okay, so where does this leave us? Well, found that there's the 3' UTRs with ISD have a 3 to 3.5-fold increase in read-through. That ISD is approximately benign, and so where does this leave us in terms of that original question I was asking, what are the consequences of translational errors? Well, it appears that one or the other selection on read-through products purges low ISD sequences, or vice versa. And so I mentioned this in the title up here, I need to bring it back to this. So, and because it's leaving behind a lot of benign non-coding variants, that could potentially provide a pool of not harmful non-coding sequences that evolution can draw from for future innovations, which might help explain phenomena like de novo genes. And that's it. I'd like to thank my advisor, Joanna Mussel, and she's giving access to stick around, and the rest of the lab, and my collaborators, and my funding sources. And thank you very much.
Could you repeat the question?
So the question is whether there's a correlation between whether a gene is highly expressed, and it's, I think, read-through rates and its ISD, both. I did look at this exact same thing where I tried controlling for expression, and I can tell you that it didn't change the results.
There was a question here.
Yes.
There are times when there are real functional genes that are based upon read-through.
Yes.
And the question is, if these were, if your hypothesis about the evolutionary history were correct, you would expect there would be certain residual properties of these active proteins that are based on read-throughs. So ones that are not sort of accidental read-throughs, but purposeful read-throughs, do they have any interesting properties distinct from other proteins?
Okay, so the question was, do proteins that have intentional read-through, I'm assuming like the side prime east, do those have different properties as opposed to other
Proteins that are not based on read-through.
other proteins that are not based on read-through? And I think that's a really interesting question, but I haven't looked at that specifically. I've been looking mostly just generally throughout the genome, so I don't know. Yes.
So, of course, biochemists like to tag things so that they can do their experiments, and sometimes that changes the function and goes horribly wrong, right? So have you thought about trying to find, and people report this in the literature, especially for yeast, right? Have you thought about trying to find those in your data set, or maybe even trying to go back the other way and see if you can use your data to predict which proteins you really should not tag?
Okay, so I think the question is using the results I have here to see which proteins shouldn't be tagged with
Whatever epitope.
with whatever. I haven't actually looked at that, so I don't know.
We still have time, so if there's more questions.
Really fast. Yes.
So, have you looked at the proportion of reads that are in frame with coding sequences that are present in the 200 years versus?
That is kind